Good afternoon, students. In our last class, we learned about the diffusion, osmosis, osmotic pressure, etc. Today, we are going to learn more terms in this chapter. Then, first is the tonicity. Tonicity is the relative concentration of the solution that determine the direction and extent of diffusion is called tonicity. Relative concentration meaning if we compare or relate it with the cell sap, the content of the cell, if we compare a solution that is determining how fast the osmosis takes place or diffusion takes place, then to which direction the diffusion and osmosis takes place is determined by the concentration of the solution, that is the tonicity. There are three kinds of solution, that are isotonic, hypotonic and hypertonic solutions are there. Isotonic solution meaning if the concentration of the solution is same as the content of the cell or same as the cell sap, such a solution is known as isotonic solution. If we put the cell in an isotonic solution, there will not be any osmosis. Osmosis will not take place. That is, water will not enter into the cell or water will not lost from the cell. Such a solution is known as isotonic, same concentration as the cell sap. Then the second kind of solution is known as hypotonic solution. Hypotonic meaning less concentrated than the cell sap. The solution outside the cell will be less concentrated than the cell sap. Such a solution is known as hypotonic solution. If we put a cell in hypotonic solution or less concentrated solution, the water from the solution will enter into the cell and cell will absorbing the water, the cell will swells or enlarge, that will happen. Or we can see that endosmosis will take place. Endosmosis meaning water from outside enter into the cell. Entering into the cell is endosmosis. Then another kind of solution is known as hypertonic solution. Hypertonic solution meaning the solution outside the cell will be high concentrated or water will be less in high concentrated solution. Hypertonic solution meaning salt concentration in the solution will be high in the outside solution. If we put a cell in hypertonic solution, exosmosis will take place. That is, water from the cell will go out of the cell. So the cell will become shrink. That will happen. Then result may be as follows. Here you can see in case A, the cell shape and size remain unchanged if we put in the isotonic solution. If we put the cell in hypertonic solution, cell shrink in size and loses its shape. Then C, if cell slightly enlarges or even burst. That is what will happen. Then again, another important thing you have to notice here. In case of plant cell, another striking feature determines the behavior of the cell when subject to varying external fluid environment. This feature is the rigidity of the cell wall. We know that plant cell will have a cell wall around it. So it will resist the bulging. If the cell absorb water and try to enlarge, that cell wall will exert a pressure on the cell content, so it will resist the bulging and protect the delicate cellular part inside. So that condition of the cell is known as turgidity. That behavior of the cell is turgidity, that condition. Then again, sometimes the cell content lose its water content. That process is known as plasmolysis. cell shrink, that process is plasmolysis, and that condition of the cell is flaccidity. Here you can see in the picture, you can see the what will happen. Two cells are taken here. One is the animal cell, that is the red blood cell, RBC is taken here. Then that is put in isotonic solution. In case A, it is isotonic solution. Animal cell and plant cell is taken here. So if we put the plant cell or animal cell, here RBC is shown here, no entry of water or no loss of water, no loss or gain of water by the cell. So the cell will be in the same state. Then in the second case B, the cell is put in hypertonic solution. Then water entering the cell is less than the water leaving it. So exosmosis takes place. That is, water inside the cell will be absorbed by the solution outside, hypertonic solution. So in the RBC you can see the cell is in a shrunken stage. Then again in plant cell you can see the cell wall is maintaining its shape. That square shape is maintained by the cell wall, but cell membrane and the all other content is shrink here in the plant cell. That is because of the exosmosis. Then in case of C, you can see the RBC absorbing water, it has only a cell membrane outside it. So it bulge or enlarge. After that, that cell membrane cannot hold the pressure of water inside it, so it is bursting. In the case of plant cell, we have seen that bulging is prevented by the cell wall around it. So the cell is in a turgid condition. That means the fully extended condition when it is put in hypotonic solution. Then another phenomena you can see is the active transport. Active transport, a new term known as active transport. Active transport meaning the cell sometimes using the energy absorb certain things or salt or ions from outside. That cell against the concentration gradient from that means the concentration of the cell sap will be higher because of the presence of ions and salt. Then the soil water may not soil water may be hypotonic compared to the cell sap. By using some energy, the cell is absorbing the ions and salt from the soil. That kind of absorption is known as active transport. Here you can see its explanation. Certain nutrients such as ions of nitrate, sulfate, potassium, zinc, manganese, etc. cannot pass through the cell membrane of the root cell easily. This is because of the concentration is higher inside the root cell, and it is so maintained in order to develop osmotic pressure. For the osmosis, plants are usually absorbing more water by osmosis. For keeping the osmotic pressure, that salt concentration must be maintained by the root hair. Then in this way, the concentration gradient of these ions is opposite to that of diffusion. In diffusion, we know that water molecule, only water molecule move from its higher concentration. Water molecule, its concentration will be more in outside area. That may be diffused into the cell through the cell membrane. But the salt are absorbed by active transport. Forcibly it is absorbed by the root hairs. That using the energy in the form of ATP. We know that by the oxidation of glucose, mitochondria is storing the energy in the form of ATP. Using that ATP, using that energy, roots are absorbing nutrients like the ions and salt from soil using the energy. That is active transport.